This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 161, recorded on September 14th, 2017. This episode is brought to you by the Defense Threat Reduction Agency, part of the U.S. Department of Defense. The agency's Chemical and Biological Technologies Department hosts the 2017 Chemical and Biological Defense Science and Technology Conference to exchange information on the latest and most dynamic developments for countering chemical and biological weapons of mass destruction. Find out more at cbdstconference.com. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, how are you?
I'm well, thank you. And you?
I'm fine. Things are good.
Everything is good in San Diego.
Yes, sir.
So you don't have no earthquakes, no hurricanes, nothing like that, right?
Well, I understand they're bleaching the streets in San Diego.
Really? Why would they do that?
Is it hepatitis A, Elio?
Hepatitis A is rampant here. It's a problem.
Right. Yeah. Oh, I heard that, yes.
We have a large homeless population, and they are susceptible.
Yeah.
Yeah,
interesting.
It's a problem.
I heard about that, yeah. And that voice, of course, is from Ann Arbor, Michigan, Michele Swanson.
Hello.
How are you?
Fine.
Did your football team lose? Is that the problem?
No, they won.
Okay, but then you should be great, right?
Well, yeah. That's a small part of my life, but it's
I thought it was 90%. Just kidding. Good to have you. Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Who you weathered
Irma unleashed the last of her wrath on us Monday, and it lasted from about 3 a.m. all the way to about 7 o'clock at night. And it dumped a lot of rain, and the wind gusts were up to about 75 miles an hour at one point in time. And it shook the house, and I almost had waterfront property there a few times, so it was pretty spooky at high tide.
Well, good. So, glad to see you're okay, or glad to hear it.
Yeah, the university had to move a surgeon from the main hospital over to the new hospital, and they actually moved him by John boat. He had to do an emergency surgery. So our public safety people had a boat, and there was 5 feet of water on the street in front of the university. So it was very nautical, is what everyone was saying.
Well, glad it's over, and glad.
Oh, I'm glad
okay,
it's over.
Not too much. We're going to talk a little bit about Houston today, and they have some problems still there. I want to tell you first about an opportunity. If you've ever dreamed of working at the Smithsonian, now's your chance. The American Society for Microbiology has an immediate opening for a public outreach fellow who will work closely with ASM and the Smithsonian's National Museum of Natural History to develop public programming supporting an exhibit on viral zoonotic diseases. This is a 12-month paid position with benefits. Perfect stepping stone for someone looking to break into the world of science outreach. You can learn more and apply at bit.ly/ASM fellowship.
Boy, if I were young.
You would do that?
Right.
Absolutely.
Sounds very cool.
Yeah, this is an exhibit that's going to be on May of 2018, and it's collaboration with ASM, so it's a great opportunity to learn how to do these things. All right, I want to talk a little bit about Harvey in Houston. We got an email from Anthony who wrote, will this be on Twix? He sent a link to an article in the Washington Post, and the article was written by Ben Guarino. It was posted online September 1st, and the title is The Health Dangers from Hurricane Harvey's Floods and Houston's Chemical Plants. And Anthony writes, I recognize Dr. Hotez's name from TWiP. And one of the scientists quoted in this article is Peter Hotez, who many of you should know, and is a well-known parasitologist, currently at the Sabin Vaccine Institute. And Anthony writes that he heard his name from TWiP. And, of course, we had Peter on TWiP, TWiP 29. We talked with him. Anthony writes, I've read that the biblical plagues of Egypt were based on an actual flood and its disease aftermath. So I thought we would talk a little bit about this. There's also an article in the New York Times, which I'll get to. But this one in the Washington Post talks about the flooding and its aftermath. This is, of course, in Houston, Texas. Peter Hotez is quoted early on saying, "I distill it down to short-term, long-term, and big picture." And Peter Hotez is not only at the Sabin Vaccine Institute, but he's the dean of the National School of Tropical Medicine at Baylor College of Medicine in Houston. And this article covers some of the after effects of Harvey. Of course, people who die by drowning in the floods. They also mention that floodwaters can draw out unwelcome wild animals like snakes, fire ants clumped together in floating rafts. In Florida, you can get alligators in your house. But
we had alligators here in South Carolina.
And did you have any in your house?
No, they were in 2 subdivisions over.
Yeah, they come out.
Wow.
But in this article, they talk about short-term effects, infectious diseases. And that's because sewage systems, as they write in the article, sewage systems spill their guts, and the water can dredge up things more disturbing. In New Orleans in 2005, the flooding from Katrina exhumed corpses, sending coffins afloat through neighborhoods. It's not easy to predict the nasty microbes that will strike. We don't have enough epidemiological studies, Hotez said. But Katrina, which hit land the same time of the year as Harvey, could offer lessons. In September 2005, the CDC reported 30 cases of MRSA, an antibiotic-resistant staphylococcus, among a group of New Orleans evacuees sent to Dallas. And Vibrio pathogens sickened 24 people and killed 6. There were also reports of skin rashes from infection and heat, as well as red marks blamed on biting mites. And they ask in this article, could we have an outbreak of cholera? And Hotez says, certainly the conditions in Houston could promote cholera, but you'd have to have somebody infected with cholera coming into the area. Stress also jeopardizes immune system, hard to maintain food hygiene in disaster zones. Katrina unleashed gut diseases triggered by E. coli and a lack of safe food and potable water. And during Katrina, there were respiratory infections among people in shelters, including an apparent uptick in tuberculosis. There are also chemical dangers in Houston. There are refineries that have exploded and spilled chemicals. There are drug shortages because, all the movement of people makes it hard to get drugs. And if that includes antibiotics, that's a problem as well. And mosquitoes. Based on experience following Katrina, there will be several competing effects on the population of mosquitoes and the prevalence of arboviruses like dengue. Well, the arboviruses can carry viruses like Zika virus, dengue virus, and West Nile. Mosquitoes need stagnant water to lay eggs. Winds and flood will wash away containers that would have been breeding pools. And so Hotez anticipates that in the immediate future, local mosquito populations will decline. But once floodwaters recede, mosquitoes will recover. In 2006, a year after Katrina, Tulane University reported that cases of West Nile infection increased more than twofold in communities that had been in the hurricane's past. And they said that the authors of that study suggested increased exposure was the culprit. Fleeing partially submerged building, people spent days waiting outside for a rescue. There's going to be a need for insect repellent down there because people are outside when places without air conditioning, you leave the windows open and so forth. And then one long-term problem is mold. Mold is a hurricane holdover. This individual, Hayden, who is
the gift that keeps on giving.
It keeps on giving.
Oh, yes.
So after Ike, which hit Galveston, evacuees couldn't get back to their homes for 2 or 3 weeks. And in a waterlogged, overheated home, mold can run rampant. The Washington Post reported that 2 months after Katrina, CDC found mold in the walls of half of 112 water-damaged homes. Where the worst symptoms are typical allergic reactions, rarely fatal, but can exacerbate other health problems. This is interesting. Post-Katrina mold was implicated in the deaths of 4 Southern University New Orleans professors, all of whom worked in the same storm-damaged building. They all died within a few months of each other.
Wow. Do they say what mold it was?
They do not.
Mm-hmm.
Aspergillus fumigatus is a good candidate.
I was going to guess it was Aspergillus.
Let's
see. It can get invasive.
Or Stachybotrys. Stachybotrys is a big one down south, and it's pretty nasty.
Yes.
And it can penetrate into deeper tissues.
Mm-hmm.
Mm-hmm.
I'm looking at the original article, and they're not identifying it.
I bet you Joan Bennett might know.
Oh, sure. Sophia.
When I was talking with Joan at the ASM microbe meeting that was in New Orleans this past June, she was regaling horror stories of Katrina and the mold bloom that occurred in her own personal home because it was, of course, flooded and damaged. And Joan was just saying the fungi were just out of control and growing everywhere. And she was still having some allergic sensitivity. She says, and the mold is high still because she was coughing and wheezing, and she maintained it was from the spores that are still resident in New Orleans, even now, all these years later, 12 years.
Sure.
So that reminds me, Joan and another microbiology colleague of ours, Rita Caldwell, are two of the leads on a new report from the National Academy of Sciences on microbiology of the built environment. And they discuss many issues and basically lay out a roadmap for where we need to go with our research effort and our policies to address these problems. And obviously with the flooding in Houston and the hurricane in the southeast, these issues are going to come to the fore.
That's a publicly available document, isn't it?
It is, and yeah, we can link to it.
So it should go on the show notes.
Yep, we will do that.
And there's also the American Academy's publication on microbiology of the built environment that Joan was one of the organizers of. That's available as well.
That's right. We'll do both.
So they found Stachybotrys. Did you mention that, Michael?
That's the one.
And they said
That's the one.
Yeah. At very high levels in the building where these professors worked. But if that did it or not, we don't know. I also found this week, published September 11th, an article in the New York Times by Sheila Kaplan and Jack Healy entitled, Houston's Floodwaters Are Tainted, Testing Shows. And the New York Times organized some water tests, and they found that water flowing down Briar Hills Parkway in the Houston Energy Corridor contained E. coli, a measure of fecal contamination, at a level more than 4 times that considered safe.
Oops.
And in the Clayton Homes public housing development downtown.
That's not too much, actually. I'm surprised.
Yeah.
4 times sounds like a lot, but when it comes to bacteria, and we're talking about the huge numbers of bacteria that exist in the environment, 4 times does not strike me as particularly threatening.
Well, if we read on, one home had level 135 times higher than those considered safe. So
it's more like
yeah.
What does anyone know when they close a beach? What's the level of coliform count that they use?
I don't.
It's pretty low. I know that.
Yeah, I'd be curious if it's 4 times or 10 times or what. Yeah, this is one family's living room. There was standing water in it, 135 times. So there, you must assume that the sewage backed up, right? Came out of the sewage system and contaminated the room.
Of course, you have to ingest it.
We have to ingest it. That's right. You're not likely to be ingesting
if you're careful
that water.
about boiling your water or using bottled water, you're not going to get into trouble from that.
There was one quote in this article. Here we go. Everyone has to consider the floodwater contaminated. He also warned residents, this is Dr. Percy, to avoid letting cuts and scrapes come into contact with floodwaters, which can cause an infection. This one individual developed scabby infections on his legs where rain boots had irritated his skin. Went to an emergency room, was given antibiotics. Mr. Greer said swimming pools around his neighborhood are rank. All the pools are just giant toilets you're unable to flush.
Oh.
Well, that sewage flowing into them, right, has nowhere to go. Anything else?
What I found really interesting about this is that we think about citizen science, but this was actually New York Times organized this research effort. So they recruited faculty from Baylor Medical College and Rice University. They partnered with the Houston Health Department, and they paid for a commercial lab, the A&B lab, to actually do the analysis.
Yep.
So these investigative reporters actually did, wet lab work to try to get to the bottom of this and alert the public of the risk.
I thought that was interesting, yeah.
Really interesting. Good for them.
Winfried Hamilton, director of the Environmental Health Service at Baylor, said, and a member of the test group that was organized, as Michele said, said she is especially worried about exposure to mold among people who are moving back or who bring their children along. as they try and clean and repair their houses. I'd be wearing a mask with a filter, she said, and goggles and gloves with rubber boots. I would change my clothes immediately after leaving the house, put them in the wash with nothing else. Mold is taking off all over the city. People with allergies or asthma are particularly sensitive to it. If people have bad headaches, respiratory problems, swelling of a limb, or a bad rash, go see a doctor right away. Don't assume it will go away on its own. She also encouraged people to keep an eye on their kids. We have a lot of what looks like sand, like something you might want to make a castle of, but it's not clean sand. This is sludge sediment.
Ooh.
Besides E. coli, what else would you worry about?
Enterococci.
Yeah?
Enterococci.
Oh, yeah.
Enterococci is another water criteria that is often used for recreational water quality. So enterococci and E. coli are indicative of, contamination from sewage treatment plants or just people defecating. And when I learned water microbiology once upon a time, you could actually tell if it was a bear, a mink, a chimpanzee, or a human based on the fecal coliform ratio.
Mm-hmm.
And you would determine the ratio of coliforms to enterococci, and you could tell who was going upstream and soiling your water.
How about Acinetobacter? Is that waterborne, Michael?
Typically not. It's typically it hangs out in ceiling tiles mostly.
In what?
But ceiling tiles. It loves cellulose. Loves cellulose. But it's a garden variety gram negative, so it's prone to desiccation. So it makes slime as well, and it hides pretty well. But remember, it's a
nasty bug.
It's a nasty bug. And remember the Acinetobacter outbreaks that the U.S. military had in Iraq, a relatively desert-like environment. So it likes water just fine, but it can actually tolerate drying better than most of the typical water pathogens.
The other group I'd worry about is all the mosquito-borne viruses.
Mm-hmm, for sure.
I think there'll be a delay before we hear about those.
That's correct, yeah. Yep.
Oh, they talked about West Nile already.
Yeah, they might. So, as soon as the floodwaters recede and the mosquitoes reestablish their breeding areas, you could see increases for sure. Yeah. So hurricanes do a number of things, and alterations in the microbiology.
It shows you how fragile our environment is.
Quite. Yeah, quite.
And it's especially striking in Houston because that is really a modern, really muscular city. And it was brought to its knees by this flooding.
Flooding will do that, right? So many feet of water, just you can't live, you can't get anywhere, you can't work. It's a real problem, yeah.
Well, it also doesn't flow because, if it floods in and then the floodwaters recede, there's no place. It's not like it's a natural stream where the water goes in and where the water goes out. It's sort of stuck.
Like New Orleans, where it could recede into the ocean, right?
Well, New Orleans is also below sea level, so it actually just sits there, and that's part of the scare. That's what we had here in Charleston this week is the water from the ocean was breaching the seawall in front of the famous battery.
Right.
And the water was crashing over, and it was literally on the streets. So what the city did is they brought in 3 gigantic pumps, and they began to pump out the sewers and pump it out into the sanitary sewer to get it off of the streets to effectively combat that. And we were fortunate that we had the pumps, and Charleston is so low that they planned for it, and fortunately their planning paid off.
Well, this is these individuals obviously need help, and federal relief should come automatically. Should not be a political issue, which it unfortunately does become. But we wish all these individuals the best in getting their lives back together again. Lots of things to overcome.
So I found the numbers for total coliforms. The single sample standards are you can have up to 10,000 organisms per 100 milliliters of sample. For fecal coliforms, of which E. coli is a member, you can only have 400 organisms per 100 milliliter sample. And for things like enterococci, which is a gram-positive microbe, the standard for beaches is about 104 organisms per 100 milliliter sample. And your fecal to total coliform ratio should be greater than 1,000 total coliforms if the ratio exceeds 0.1.
Wow.
So
these are very small numbers, really. These are stringent criteria, don't you think?
Yeah.
Yeah.
So the fourfold in Houston would close a beach for sure.
Yeah.
Yeah.
And the things that the TWiM users will send you an email about is, of course, we have to worry about Cryptosporidium and Giardia. Cryptosporidium, of course, crippled the Milwaukee water system a few years back.
Yep.
And Giardia lambia is still crippling the St. Petersburg water system in Russia, because you can
Luckily, these are not marine organisms. So insofar as the flood is caused by the surge, that would not be in play.
They're not going to be in there. They're not going to be in there.
Yeah. Well, thank you for getting that info, Michael.
Google is a wonderful thing.
It is. It is. This episode is sponsored by the Defense Threat Reduction Agency. Imagine an everyday, inexpensive drone you could buy online, modified by terrorists to spread chemical or biological weapons over a crowded football stadium or holiday parade, plague, VX, sarin, weaponized flu. How could we prevent a scenario like this from happening? How would we treat the victims? How could we counteract the effects? Join us in Long Beach, California, November 28th through 30th for the 2017 Chemical and Biological Defense Science and Technology Conference to exchange information on the latest and most dynamic developments for countering chemical and biological weapons of mass destruction. Collaborate with more than 1,500 scientists, subject matter experts, military service members, industry partners, and academic leaders from across the globe who are committed to making the world safer by confronting chem-bio defense challenges. Part of the U.S. Department of Defense and charged with safeguarding our warfighters and our nation, the Defense Threat Reduction Agency's Chemical and Biological Technologies Department hosts this important conference and brings together the best and brightest from around the world. Please join us to share your important ideas. For more information and to register, you can go to cbdstconference.com, all one word. You can also find out about the latest information about the meeting either on Facebook or on Twitter. On Facebook, just search for DODDTRA, all one word. And on Twitter, the handle is CBDST Conference. The 2017 CBDST Conference, today's innovation, tomorrow's warfighter capabilities. Now we have a paper for you, which should be on Valentine's Day, but it's not. Right, Elio?
Absolutely. It's about sex. My God. Got your attention, didn't I?
You did.
It's going to drive people to download it.
You bet.
Okay. And the title is Mating in the Closest Living Relatives of Animals is Induced by a Bacterial Chondroitinase. I'll explain all that. It's by Ariel Wojnicka, Joseph Gerdy, Ryan Hewitt, John Clardy, and Nicole King. I recognize the last name. Nicole King is a distinguished scientist in trying to study the development of multicellularity, among other things. This paper appeared in Cell in September, and it's brief but to the point. So here's the story. First of all, we know that sex is kindled by many factors, but not all of them are expected. So to the novel ones is the fact that bacteria make aphrodisiacs. Sorry to disappoint you, they're not aphrodisiacs that work on people. They work on a very tiny...
That you know
I know of. They work on a little organism which is supposed to be as close as they come to the origin of multicellularity, and they're called choanoflagellates, CHOANO flagellates, one word, which, first of all, let me say, in context, the fact that bacteria talk to other forms of life is the rule now. This is a relatively novel thought in the sense that it was always there since the very beginning of pathogenesis studies, but the idea that it's pervasive and that all bacteria practically talk to somebody else, and they talk to themselves, they talk to other organisms, that is really almost a paradigm shift. In other words, microbiology is a social science. You agree?
Totally.
Okay.
Yes.
Now, let me say that for those who are interested in a phenomenally wonderful review There is one written by McFarlane Guy and colleagues, and we'll put that in the, that's some years ago. We'll put that in the show notes, okay?
Yep.
Anyhow, solitary existence in microbiology is limited to petri dishes and maybe to some infected hosts. The rest are all in communication. So the labs of King and Clardy found something surprising. In fact, their summary starts by saying, we serendipitously discovered. What did they discover? That an ordinary marine bacterium, Vibrio fischeri, the guy who's involved in bioluminescence and interacts with the bobtail squid that McFarlane Guy studied, the one that we talked about. That organism, when you put it together with these, I hate to call them primitive animals, but so these ancestors or related to the ancestors of animals, the choanoflagellates, induce a sexual frenzy, a mating frenzy. These guys are able to reproduce sexually. That was known because they have the genes involved. However, if you want to do that in the lab, you got problems. You got to starve them for several weeks in a row. Then a very, very few deign themselves to undergo mating. So this is really interesting because suddenly you add bacteria and bang, within 30 minutes they are at it. They make swarms, that is, they mix cells come together in swarms of about 30. A couple of dozen anyhow, and they do this within 30 minutes, and this is followed by sexual reproduction, cell fusion, karyogamy, that is the fusion of nuclei, meiosis, recombination. And they study all that. They show that all those things take place, which is very good because just showing mating, that is cells coming together, does not mean that sexual reproduction ensues. But here they did a rather careful study, which, by the way, is handy because these choanoflagellates need to be studied more, and everything that you add to the knowledge. So anyhow, this is news, and what is nice is that this happens when you add as few as 10 to the second, that is 400 cells per ml, which is powerful. So this is few bacteria. We already heard that that's a number that would close a beach, but in the context of the kind of thing that we talk about, this is a very small number. And this is comparable to the concentration of Vibrio fischeri in the ocean. By the way, they are not pathogenic to humans, these Vibrios. How did they find this out? Well, this is a nice story. They were looking to add a control to an experiment where they were going to use some other bacteria in connection with the choanoflagellates. And they said, well, what is most innocuous? What could be more of a control than Vibrio fischeri? And guess what? They found this frenzy going on after their addition. So no wonder this is serendipity, and they must have really said, what is going on here? All right. So what is the aphrodisiac that these bacteria emit? They can show that this is a compound found in the medium where the bacteria grow. They can use spent medium that is filter the bacteria out and add it to the choanos, and sure enough, they go into this behavioral change. Okay, so they can use this as a bioassay to purify the principle. So what is the aphrodisiac? You would think it's a small molecule or a hormone or something like that. Guess what? It's not. It's a 90 kD protein, which they call EROS. Subtle, isn't it?
Isn't that great?
Eros. I love that.
It stands for extracellular regulator of sex.
It's great.
So what is Eros?
They must have stayed up nights to come up with that.
It doesn't take much. In fact, it works at 5 picomolar, which is pretty powerful as aphrodisiacs go. So this is not your regular homegrown aphrodisiac that may or may not induce behavioral change. This is good stuff. Okay. So once added, this is what happens. There's sexual reproduction ensues. So what is Eros? What kind of a protein is it? Well, it's an enzyme. And it's an enzyme that hydrolyzes chondroitin sulfate. The listeners who've had biochemistry will remember what chondroitin sulfate is. But for those who haven't, let me tell you, it's a glycan. It is a sugar polymer. It's a polymer of glucosamine and glucuronic acid. And it's found on cell surfaces and on the extracellular matrix of animals, where it's involved in differentiation and in cell proliferation. By the way, chondroitin sulfate is also used medically for those who have bad knees. And I'm not making an endorsement out of it because I have no idea if it works or doesn't. It's a little bit difficult to see how a large molecule like that could penetrate, but apparently there are those who believe it. Anyhow, so in the cells or in the tissues, it's linked to proteins. So this is a glycoprotein. Now, this is about the story. There's not a tremendous amount more to it, except to tell you that they believe that this modification of chondroitin sulfate by Eros is not just adding or supplying sugars, because when the chondroitin sulfatase works, it converts chondroitin sulfate into sugars, disaccharides mainly. But if they take those disaccharides, the products of hydrolysis of chondroitin sulfate, and add it, nothing happens. So they think that what happens is that the hydrolase works somehow by modifying the structure of chondroitin sulfate. Now, what chondroitin sulfate does on these beasts, by the way, I didn't tell you, these guys look like sperm cells. They have a head and a long tail, which is what makes them flagellates. So, and when they mate, they make head-on. That is, their heads touch. Anyhow, so what is going on here is interesting, and it works, as I say, at very low concentrations, both of the bacteria and of their product, Eros.
Elio,
Now,
could I interject? You mentioned that they look like sperm cells. JP, one of the authors on the paper, pointed out that sperm cells also make an enzyme like this that they use to digest the target egg.
Huh.
And it's
So
got
penetrate.
I missed that.
Yeah.
Oh,
Yeah.
my goodness.
Yes, there's a lovely parallel. Yeah, it's not in the paper, but I got that from chatting with JP.
I see. No, oh, wow. That's great. No, I had missed that, but I missed it because it wasn't there.
Right.
Anyhow. Yeah, and they sort of suggest that this is possibly a modification of the compound on the surface of the choanose, which makes them capable of mating.
Yeah, you could imagine that it makes them sticky, right? Taking off the sugar does that.
That's right.
By the way, I don't know if you mentioned this, but they made a single amino acid change to destroy the catalytic activity of this enzyme, Eros, and that kills the aphrodisiac properties of it.
Right.
Which I think is important.
But that doesn't mean that it's the enzyme activity itself that, per se, that in terms of making products that are bioactive. It simply says that what they suggest is that it's a structural change.
Sure. That
you modify the
substrate, right?
sulfate sufficiently that it allows this behavior. So, you want to add anything else before I'm close to wrapping this up, and this is early.
I was really taken by their beautiful meiosis data. They wanted to demonstrate that they really did recombine, which is a hallmark of fusion. And so they did a genetic test, and they show us a lovely graphic where we look at the recombination between two different alleles of the donor and the recipient, or the two partners. And it's laid out really beautifully graphically. And they had that data. They knew that meiosis was happening before they were able to capture it by video. So it's just it's really compelling complementary data sets.
A really lovely movie, so you can see the aggregation, the swarming and aggregation. But yes, you're right. This is a nice, very thorough piece of work. But I think the motivation in part is because these labs are very interested in the choanoflagellates.
Yes.
And so in a way, I wouldn't say this was an excuse, but it was a good occasion to go further into the biology of the choanoflagellates.
So let me add that we had Nicole King on TWiVo not too long ago.
Did you?
It was TWiVo number 11,
And remind us that acronym means
This Week in Evolution. That is a podcast I do with Nels Elde at the University of Utah.
Right.
And Dr. King was our guest for number 11 when she talked about, so she works on these choanoflagellates, and up till now, she has worked on the fact that they form multicellular rosettes, and that this is triggered by lipids produced by an environmental bacterium, Algoriphagus machipongonensis. How about
that's
that?
Right.
And so
I'm glad you mentioned that.
And that's what she worked on. That's what she described on TWiM. And it's a lovely episode. And then, as Elio said, they were looking for a control, and they stumbled into this effect of Vibrio supernatant. So I think it's a lovely example of serendipity. The fact that they got one protein that has the activity is amazing, right? If it had been more than one, it would have been hard to pick up. And I also like that they mentioned in the discussion they have since identified other bacteria that similarly induce swarming and mating.
That's right.
So
they're all Vibrios, by the way, but not all Vibrios do that. So it's not just being a Vibrio. So some Vibrios do and some Vibrios don't. So
I think it's really interesting, and it's probably just the tip of the iceberg.
It is, and they promise to look further into it, and I think they will.
I'm sure.
And I think they're doomed to succeed.
Doomed to succeed.
Doomed to succeed.
Very good.
My prediction is they're gonna get there. They're gonna find more cases like this.
So I had the pleasure of talking with both of the first authors. Ariel Woznicka was a PhD student in Nicole King's lab, and she originally was really interested in chemistry, and in particular sphingolipids, and she was just convinced that these were very powerful molecules, and she wanted to understand them as a chemist. But then when she was a freshman, she took a class on cancer biology, and Professor Joyce Schroeder showed a movie of moving cancer cells. And Ariel says she was just floored by this video. It just found it incredibly beautiful and fascinating. And she determined right then and there that she wanted to be a part of this. She started emailing professors looking for a place to work and ended up with a research lab as a freshman. So what she really loved about Nicole's lab is that she encourages people to follow their curiosity and follow your gut and really pursue this fascinating biology. And as you point out, they are convinced that these choanoflagellates are a great experimental model. And right now, it's still a relatively small field. About 50 people might come to their meetings, their conferences. So as you pointed out, they really do feel like they need to do the work and really get the beautiful demonstrations of the biology. So Ariel was pointing out that they had the meiotic data. They knew that these tiny little fast-moving creatures were mating, but they wanted to capture it in video. And she said that was something that took a lot of painstaking work because only about 25% of the population mates. They're very small. So she persisted and was really pleased to get these beautiful images and movies that are you can link to them in the paper.
Most of the cells mate, don't they?
I thought she said about 25% of them
that sounds about right, Michele.
Under the conditions she was looking. She also credits Nicole. She said that she, as you all pointed out, she was setting up to do this other experiment. She noticed these clumps. She tracked the kinetics, and she was really convinced of it. She brought her advisor, Nicole, in to see it. And after looking through the microscope, Nicole said, "They're mating." And Ariel was really surprised that she had such confidence, but sure enough, after they went through the experiments, she was right. So it's just been a really dream project for Ariel, and she, Vincent, you'll be pleased to know, has moved on to a postdoc with Julie Pfeiffer.
Oh, cool.
Where she's going to be looking at viruses in the gut and the impact of the microbiome on the viruses. So she thoroughly enjoyed this collaboration with the Clardy Lab. The two labs have already published 5 papers together since 2012, and Ariel was a co-author on one of those papers before. So they've got this well-oiled machine worked out between the lab at Berkeley and the lab at Harvard, which is where the other first author was, J.P. Garrett. He says that as a kid, he was always curious and asking lots of questions. He got his start really thinking as an organic chemist and loved that class. But then when he was taking a molecular biology class, he kind of made the connection between living chemistry and decided that molecules that control life and developmental biology would be a great place to focus. So he got his first lab experience as a freshman in college and then continued on that path. He did his undergraduate work at University of Illinois, Champaign, and then his PhD was at Madison studying quorum sensing in Pseudomonas. So studying a well-characterized homoserine lactone and its impact on behavior of Pseudomonas. But he really loved the idea of joining Clardy's lab because he wanted to study some new signaling molecule that had never been characterized. So kind of break new ground in this field. So it was a collaboration that was a dream come true. He describes one especially exciting moment was when he was trying to purify the active aphrodisiac. He finally got a clean single band. He eluted it, added it to the cells, and looked in the microscope and sure enough saw that they were the mating morphology. So he called some other people in the lab to come look. He blinded the experiment and then convinced himself that, he had the molecule. So then they did the mass spec, and when he found that it was a enzyme that breaks down glucosaminoglycans, he started doing the Google searches and then realized that sperm cells do something similar. So that was a really exciting period. So he's continuing some of this work, but is also working on a separate project looking at other signals that control a different microbe, Plasmodium falciparum, so the agent that causes malaria. So he's got some interesting biology. Stay tuned. That might be coming to a journal near you soon.
Nice.
They each had some advice for other scientists, especially junior colleagues. JP said to be sure to keep reading and asking questions, talk to other people. It's where you can get a lot of ideas and inspiration. And
listen to This Week in Microbiology.
That's right. And Ariel also said, curiosity, don't be afraid to ask questions, trust your data, pursue the unknown. And she was really fortunate to be in Nicole's lab and now Julie's lab, where they really encourage their trainees to be bold and break new ground. And they were both quite excited about their paper being featured on This Week in Microbiology.
Wonderful.
Oh, good.
Thank you. Thank you, Michele.
Yeah, my pleasure.
Elio, I have a question for you. How could one show that this aphrodisiac actually functions in nature?
Oh, boy. Well, assume that you can find an antibody, and you add an antibody to a drop of seawater containing all this. That may work it, or a chemical that inhibits the action of the enzyme. I don't know what else to tell you. Those are the standard ways of thinking. People use what they call microcosms in studying marine life.
Right,
that is, they select a couple of gallons of seawater and sequester it in a bottle, and that becomes a microcosm where you can do experiments. And so maybe something like that could be done.
I remember in our conversation with Nicole, that was something she was trying to figure out for the aggregation phenotype, caused by a bacteria. How could we study environmental samples and show that this is happening? It's the same question applies here. Michael, sorry.
I was going to say, since we know they're now undergoing meiotic division, and they showed us with their genetic model how the genes are indeed exchanging amongst two distinct individuals, I would do a before and after of Elio's suggestion of an antibody against this protein, and ask the question, do you see a higher level of recombination? Because it's old bacterial genetics. If you remember back to the halcyon days of the bacterial genetics, people would look for particular markers to demonstrate transduction or conjugation. They would just look for a marker that would move. But today, in the era of PCR, that's not necessary, and you have sequencing, and you can probably sequence it or make a probe that you could add a tag to that you could light up that would find a particular sequence under an appropriate level of stringency, and you could actually demonstrate that it was indeed working. And you could forecast the mating frequency and other things based on the activity of the enzyme or how much has been taken out.
Mike, you remember when we used to show plasmid maps with times on them and not base pairs?
Oh, yes. And in fact, I was debating whether to tell the medical students. I had to I introduced the medical students to transduction and conjugation. And
I think it'd be lost on them, Michael.
It is. But you tell them that E. coli can have sex for, 100 minutes, and they sort of look at you going, really? 100 minutes? Who knew?
Who knew?
A complementary approach they could take, which they actually did in this paper, is they showed that if you add exogenously the enzyme, you can stimulate the process. So that might be another way to test it out in nature.
Yeah, that's right. All right, let's do a few emails here. We have one from Roman who writes, Dear TWiM team, my name is Roman. I am a bioinformatics enthusiast from Odessa, Ukraine. I've been listening to TWiM for 1.5 years now, and recently also to TWiV. Inspired by you, I decided to record my own bioscience podcast called the Bioinformatics Chat. On the most recent 9th episode, I was joined by Michael Tesler from the American Museum of Natural History and Christopher Mason from Weill Cornell Medicine. We discussed the discrepancies in microbial diversity when measured by the 16S amplicon sequencing versus shotgun sequencing. It's a bit technical, but I thought some of you and your listeners may find it interesting. You can find it at bioinformatics.chat. Thanks for the great show. So
That's awesome.
I've listened to a couple of these, and they are indeed technical. So it is, they're talking about computational biology and the programs you use, the approaches, the statistics. And so if you like that, you'll love it. But it's a little hard to understand if you're not totally in the field. But I think it's great. I think it's great that specialists are talking to each other and recording it. That's the whole idea of these podcasts.
Yeah, and the more the merrier. Let's get more people talking science. I also think a lot of our listeners are actually, PhD level scientists. So anybody who's moving into that field or already in that field might really appreciate that discussion.
For sure. John writes, with respect to TWiM 159, that was immunophage synergy. Fascinating study. One question. You talk a lot about adaptive versus innate immunity. But is there anything peculiar about mucosal immunity in particular that could shed lights on the results? Well, so just to remind everyone, this was a study where they showed that they used a phage to treat mice that were infected with Pseudomonas. And they showed that the phage could clear the infection, but only if the mice had intact innate immunity. And innate immunity, of course, is the earliest response to foreign invaders, viruses, bacteria, fungi, etc. And so apparently the phage needs innate immunity to accomplish a clearance of the infection. The experiments were done in a time frame where adaptive immunity, production of antibodies and lymphocytes, would not play a role. Now, John wants to know if there's anything in particular about mucosal immunity. Well, I'm sure with respect to innate immunity, of course, there are differences with respect to other sites, but the approach they used, they completely knocked out innate immunity in the whole mouse, would not bias for the respiratory tract. So I'm not sure that we know anything about that. Of course, in the respiratory tract, there's what we call secretory antibodies, which aren't found at non-mucosal sites. But as I said, these experiments don't address those. Those happen much later in the immune response. So it's not really an answer to your question, but I think there are differences in the innate responses at different sites that may participate in that.
I think the principal difference is the innate system would also accelerate things by throwing in complement.
Yeah, that's right.
And, in the absence of complement, things are very different.
Yep, absolutely. All right, we have an email from Anthony. Now, apparently Anthony sent an email to Professor Marcus Egert, who is at Furtwangen University in Germany. And he published a paper about cleaning sponges. And so he wrote he published an article, Microbiome Analysis in Confocal Microscopy of Used Kitchen Sponges Reveal Massive Colonization by S. Natrobacter, Moraxilla, and Cryptosporidium species. That's a Nature paper.
Uh-oh.
Sponges, your kitchen sponges. And Anthony wants to know, what about using bleach along with dishwashing liquid when washing dishes? That's for every time the dishes are clean, not just for occasional sanitation. In addition, once a week, the sponge is replaced. I raise cage birds and started using the bleach to minimize the risk of a disease spreading from one bird to the whole group through washing the food and water cups. So he sent this email to Professor Egert, and he responded, thanks for your interest. Our study suggests that keeping and continuously cleaning a kitchen sponge for too long might lead to a microbial community inside the sponge that contains higher shares of potentially pathogenic bacteria. Classical sponge cleaning techniques, boiling in water, cleaning in a washing machine at over 60 Celsius using a solid detergent with bleach will surely lead to a significant germ reduction, which is good. However, in view of local germ densities of more than 50 billion per cubic centimeter, organized in hard-to-destroy biofilm-like structures, and since killing curves usually can be described with negative exponential functions, it's always likely that some more resistant germs will survive even the harshest treatments and grow up again to a community with a higher share of potential pathogens. Based on our study, we therefore recommend that instead of keeping your sponge for too long and cleaning it only ineffectually, one should regularly replace it, in particular, and more often when you live, work in a hygiene-sensitive area, hospital, cafeteria, or if you have ill, immunocompromised old people at home. However, after having used them in the kitchen, you might surely use older sponges in other areas of your house where hygiene is less important than in the kitchen, where food is prepared, for example, to clean the bathroom, toilet, or for garden work. Hope that helps a bit. Well, I think you got to throw them out every week. They're so cheap, right?
Yeah, when in doubt, throw it out.
Well, that's a good one.
It's a lot of trash.
You could use dish rags, instead of sponges. They're easy, in a sense, they're easy to clean because you can boil them, you can do things with them. The sponges don't work very well. But maybe cheaper also.
Michele, you don't want to add to the landfill? Is that what you're worried about?
Yeah, there's got to be another solution. Do you think we're going to have maybe one day probiotics? We pre-seed our sponges with some bacteria that provide colonization resistance.
Maybe. Well, maybe they could make sponges that don't get colonized. Period. Right?
Yeah, that don't allow biofilms to form. I know that's wishful thinking that you can keep out microbes, but
yeah, that's true. You find some will find a way, right?
Yeah.
I do keep kitchen sponges for a long time until they look funny, and then I throw them out. But it's probably not a good idea.
I usually go by the smell that I throw them in the dishwasher.
You throw them in the dishwasher? Okay.
Yeah, I put them through a dishwasher cycle.
One more from Jim. Don't know if you guys heard this, but didn't want you to miss it. It is a podcast called Paperfuges. And this is inventor Manu Prakash turns everyday materials into powerful scientific devices, from paper microscopes to a clever new mosquito tracker. From the TED Fellows stage, he demos Paperfuge, a hand-powered centrifuge inspired by a spinning toy that costs 20 cents to make and can do the work of a $1,000 machine, no electricity required. And he gives a link for the audio of this, which was a TED Fellows presentation. So I've heard about these cheap centrifuges that you can use in the field and throw them away.
What drives them?
I think
it's a twirling.
You spin. You twirl it around your head.
Like a rubber band?
Jump rope.
Let's see. It's a sort of thing where you pull the strings apart and it spins. That sort of toy. I have a Wired article also that.
But kids have as a toy.
Yeah, it's just with string.
Mm-hmm, mm-hmm.
Yeah, so basically, it looks like he's got elastics that twist and untwist, and he pulls it out and it spins, then it spins back. And 15,000 RPM, you can do plasma separations with it. I'll put a link to this Wired article. These are the sort of things that can be revolutionary in resource-poor areas, right?
It's amazing. This guy is about as inventive as anybody I know. Incredible. He just can turn anything into a doable thing in the field under very reduced conditions. Amazing.
Yeah, it's great. I admire it. All right, that is TWiM 161. You can find it at Apple Podcasts. You can find it at asm.org/TWiM. If you like what we do, subscribe on one of your devices, your phone or your tablet. That helps us to attract advertising. And if you like what we do, consider contributing. You can go to microbe.tv/contribute to find out the different ways you can do that. And if you have questions or comments, you want to tell us about your new podcast, we'd love to talk about it. Send them to twim@microbe.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
Thank you all.
Michele, are you happy that you never get a hurricane up there?
Yeah, I will admit that a lot of my Michigan friends and family have been talking the last week about how we really are pretty protected. No big earthquakes, the occasional tornado, but yeah.
Well, you do have a lot of snow, right?
Not that much.
No?
We're not near a big lake here in Ann Arbor. So, and of course it's getting warmer, so we have less snow the last several years.
Elio Schaechter is at Small Things Considered. Thank you, Elio.
My pleasure, of course.
And Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ray Ortega for his technical help. I also want to thank the sponsor of this episode, the Defense Threat Reduction Agency. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.